cell specification, we talked about there are three types of cell specification: autonomous, conditional, and syncytial. But if you break down all three of those, there's really two major factors in specification. Number one is cell-cell interactions, so the interactions that cells have with one another to start specifying one another. This is mainly included in conditional specification. And then the second way is mostly syncytial and autonomous specification, where you have a asymmetrical distribution, meaning a disproportionate or it's not equivalent throughout the cytoplasm, of either RNA or proteins or both. So you have transcription factors, which are the proteins that activate a lot of the genomics, some which will repress various genes, and we're going to talk about those today. So these are the main two types of ways that all three of these modes of specification use. Conditional is typically cell-cell interaction. Syncytial and autonomous specification use asymmetrical distribution of these RNAs as well as proteins in the cytoplasm to begin the activation of gene expression. Okay, now speaking of asymmetrical distribution, there are three modes of distribution that you need to know about. And these are done usually before fertilization. So let's go through the three. The first one is the molecules, such as the proteins, or in some cases the RNA, are bound to the cytoskeleton and passively inherited in the cytoplasm. So when the cell divides, they're put in initial places and they're bound to the cytoskeleton and they don't move from those places. So in the initial setting up of the oocyte, certain cells can distribute and put, as you're going to see with the Drosophila today, they might put it at opposite ends of the oocyte. So you have certain RNAs on this side that are bound to the cytoskeleton, certain RNAs that are on this side that are bound to the cytoskeleton, and they're not going to move from that spot. So they stay where they're put. That's one mode of how you can distribute RNA and proteins in the cytoplasm, is the cytoskeleton holds them in place. Number two, during the processes of cleavage, the molecules are transported along the cytoskeleton. You should know that microtubules, which are part of the cytoskeleton, can be used to traffic vesicles as well as molecules from one part to another. So there are times where the cytoskeleton will actively move some of these molecules around. They're not necessarily going to stay in the same place, but there are certain mechanisms in place you're going to see in the C. elegans today that starts pushing certain molecules to one side or the other based upon the fertilization event. And the third one is during mitosis, due to the nature of the mitotic spindle and the cytoskeleton, as the cells pull, sister chromatids away from one another, sometimes you get some of these RNAs and molecules that also get pulled away from one another during mitosis and then subsequent cleavage of the cell. So that's also another mechanism, is that they might be associated with the centrosome, and they'll follow these nuclei as they move to one cell or the other, and that disproportionately distributes these molecules as well. So these are the three main mechanisms. They'll either stay where they're put in the cytoskeleton, they'll be actively moved due to some type of event that occurs in fertilization, or during the actual cleavage process where mitosis is occurring, they'll be moved with the nuclei so that they're disproportionate into new cells. These are the three main ways in which you can asymmetrically distribute some of what we call the maternal factors. We call them maternal factors, these RNAs and these proteins that are put into the oocyte before even fertilization begins. So I told you in the beginning that oocytes are not universal in terms of how these molecules are distributed, and it doesn't even stay the same way that it was put together. Things start shifting around even before the first mitotic event.